Hello students, welcome back to my class. So here I am back with new video. So in today's class I shall be teaching you new topic, that is inheritance of one gene. So in my last class I had discussed few important terms related with genetics, and I hope that you all have learned those terms. So this term is going to help you to understand this chapter better. Okay, so let's start our today's class. Okay, inheritance of one gene. This concept, okay, it can be explained with monohybrid experiment. As I told you that Mendel, he had experimented with pea plant, right? So in his experiment he did monohybrid cross. In monohybrid cross he chose two parents with contrasting character. So how does this looks like? He chooses two parents, tall plant and dwarf plant. Tall plant was represented with uppercase and dwarf with lowercase, and they were cross pollinated. Okay, then from here one gametes came. Okay, and as a result this is F1 and this is P1, parent of first generation, and F1 means first filial generation. So the progeny that is obtained by the cross, okay, from the cross of parent 1 is known as first filial generation. Now next, what happened? When the progeny is obtained, it is seen that it is tall. The progeny is all tall, and here the gamete is 50% from tall plant and 50% from dwarf plant. Though both the gametes are present here from both the parents, but the character expressed is the tall character. It is, see, according to the genotype, it should be the height should be in between neither tall nor short, right? Because it possesses both the allele, both the gamete, but the result was that the progeny were all tall. Then Mendel, what he did in next, he carried out, he continued this experiment, okay, to second generation, to third generation, fourth generation, in that way. So in next, what he did, he self-pollinated this, self-pollinated with the F1 generation. So when he did this, he found that, okay, so let us see. So F1 into F1. So the F1 was TT, right? Then from here, two gametes, right? So the progeny those were obtained are this. So this is pollinated with this, they will get the gamete. And then this. Now in this case, what happened when he self-pollinated the F1 generation? Okay, now this F1 generation, this progeny will become P2, because now they are parent, right? Because now they are cross-pollinated. Now the F1 has become the parents now. So this parents, they just segregated, right? The gametes and the progeny they obtained are this. Here, what it is observed that phenotypically there were 3 tall and 1 dwarf. Correct? Could you follow this? First, what happened? Okay, I'll just show you. First, tall plant and dwarf plant was taken, right, in the first set of experiment, and from here the gametes came was 50% from each parent, and the result got is tall plant. Okay, so here 50% is received from the tall, and 50% is received from the dwarf plant. And here we can see that the character, the trait of only one parent is expressed, but according to the genotype, the height of the plant should be like average, right? Because it possesses both the traits, both the gametes of tall as well as the dwarf, but the progeny were all tall. It means that only one trait, okay, of a parents, only trait of only one parents is expressed in first generation. Then, so after this, it continue with this, okay, where it is self-pollinated with the first generation. So self-pollination means where the pollination takes place among themselves, okay, that is called as your self-pollination. So the self-pollination took place between F1 generation. So from here, you can see that the gametes get separated and it form like this, okay, and as a result, one pure, this is your homozygous. Okay, remember the term homozygous and heterozygous. Homozygous is the pure breed, okay, heterozygous which contain, or you can say heterozygous is which contain the different gametes, right, different alleles. So this one contain the pure, so this is pure tall, this is your heterozygous, and this is again your homozygous. So if we see phenotypically, then this is your tall, this is tall, this is tall, and this is dwarf. So 3:1, 3 tall and 1 dwarf, okay. And if we see it genotypically, then what happens? 1, because the genotype this is pure, right, so 1:2. Here you see this is heterozygous, right, the genotype, the makeup, the genetic makeup is different, right? So how many are there? 2 and 1. So 1:2:1. So this is also called as your phenotypic ratio and a genotypic ratio. So this one is called your phenotypic because the external appearance is considered, and the below one is genotypic ratio. Now based on this, Mendel he proposed that something is stably passing on, okay, something is stably passing on from one generation to another generation through gametes. These are gametes, and through this gametes something is passing on to the next generation, right? And he turned it as a factor, okay, and that factor now we know it as a gene. So gene is a unit of inheritance. So gene is that factor which is passing on the character from parents to their offspring. So here you can see the experiment which he carried out. So this is your first parent P1, this is called your F1, this becomes your P2, the parent 2. Now here, like I said, right, so this becomes your what, parent 2, and this is your F2, second filial generation. Okay, here let me tell you one more thing. Now coming to the first experiment, first monohybrid experiment, here we got to see that 50% of gamete came from the tall plant and 50% from the dwarf plant, but the trait expressed was the tall plant. So the tall, the character tall is called as your dominant character, and the dwarf is the recessive character. As I had explained you, anything that overpowers, okay, which expresses the trait, which expresses its character, its features, okay, is known as your dominant character. So this is the dominant trait. Tall is a dominant, and this is your recessive. I hope that this is clear to you. Now here we are talking about gametes, isn't it? Now to show that how gamete form, okay, one British scientist, okay, Reginald C. Punnett, he has introduced a square, okay, through which the gamete formation can be expressed, can be represented, okay, and that is known as your Punnett square. So let us see how Punnett square plays an important role, or how does it shows the production of gametes, okay, by the parents during the formation of zygote. And it is a graphical representation. Like I said in the very beginning, that this was the first time where biology and maths, they came together, right? The mathematical approach was taken for the first time. So here what Mendel does with this, okay, so this gamete formation was shown by this square. So first is tall and this is dwarf. So from this, what we got to know is 50% from each, right? So here the gamete came 50% from here and 50% from here, okay? So this is your F1, first filial generation, the first experiment, monohybrid experiment, okay? Then after that, again, this is self-pollinated, self-pollination. So in self-pollination, what is observed? Okay, let me just write down here. 50% then self-pollination. This is self-pollination. Here the gametes taken with this. Correct? So T, then this comes here, this is this and this. So here you can see these two are homozygous, okay, and these are heterozygous. So here 1/4 is tall, right? Then 2/4, see, this 2/4 is this, isn't it? 1/4 is pure tall. Again 1/4 is your this, correct? So from here only gamete formation were explained, okay? And gamete, it is written on a top, on a side, okay, in a row, in a column. So in this method, the gametes could be written on a top, on a side, on a row, column. I hope that this is clear to you. Now in the second square, as you can see, where self-pollination takes place, there you can see that the tall, okay, the phenotypic ratio, as I already told you, is 3:1, and the genotypic ratio is 1:2:1. So I hope that now you have understood this. So from here, okay, from this picture, so from this experiment, from what it is clear that only genotype ratio can be calculated, okay, by using mathematical probability. So this is some mathematical calculation which was used, okay, to explain the formation of gamete, and it is observed that only the genotype ratio can be calculated. But to know the genetic composition, that from where this gamete has came from in a F1 and F2 generation, the composition, okay, so it was very difficult to know with this Punnett square. So for that, to know the genotypic composition, Mendel, what he did, so to know the composition, that from where this genetic compose are coming from, okay, to the F1 generation, because see, in first generation all were tall, right? In second generation, both from both the parents 50% traits were received, right? 50% gametes were received, and we got new trait, new type of plant, that is a dwarf. It means in the second generation, even the dwarf plant has expressed its trait. It means in second generation, both plant has expressed its traits. Whether in a first generation, only one plant has expressed the trait, right? Okay, to know the composition, Mendel, he has crossed hybrid. Hybrid could be this, okay, this, so, and this. He did the, okay, he has chosen tall, okay, so tall hybrid with the parent. So this into this. So I hope that you all remember what is test cross. So test cross is what? It is a cross between hybrid and its homozygous recessive plant. So homozygous recessive plant, right? So from here the gamete got is this. So now here what we got to see is that heterozygous tall. Okay, if this is taken, then we got heterozygous tall and dwarf, right? It means 2 tall, 2 is to 2, you can say, right? And homozygous dwarf. So this is how the composition they got to know by doing test cross. And if it is capital T, if they are taking the pure tall and pure, then the result will be your T. So from here what we can say that if it is heterozygous, okay, if the hybrid, if the one of the parent is heterozygous and one of it is recessive homozygous, then the result come is 50%. Okay, 2 tall and 2 dwarf. On the other hand, if it is pure tall and pure recessive, the result will be all tall. So because the tall character is a dominant character. Are you clear with this? Okay. Now, based on this monohybrid observation, based on this monohybrid experiment, Mendel, he proposed some rules, okay, of inheritance, and those rules are now known as principle of inheritance or law of inheritance. And there are two law of inheritance based on monohybrid experiment, okay, based on monohybrid cross. So those two are law of dominance and law of segregation. Now the whole concept is clear to you, right? Now let me tell you about the law of dominance. In the first one, okay, I had told you that, okay, so the first experiment, this one here, I had explained that 50% came from both, and all the progeny here in F1 is tall. So from here what we got to know is that tall, one of the trait is dominant over the another, isn't it? So this expresses your law of dominance. So there are some features of law of dominance. So you see page number 75 from NCERT book. Could you see? Yes. So here you see in law of dominance some features are written, okay, that characters are controlled by discrete unit called factors. See, this I had explained you that there is something stable which is passing on from generation to generation, generation to generation, and that discrete unit is called as factor. And factors, they occur in pairs, right? Genes, they occur in pairs. Then the third one is that in a dissimilar pair of factors, one member of the pair dominate the other. So when I say that when the pair of the factors, okay, when they are dissimilar, like this. So out of this, the dominant, this is the dominant one or not, the dominant pair will dominate the other, the recessive. This one is recessive and this one is dominant. So these are not similar, right? So this will overpower this. Okay, so this is your law of dominance. So the statement is that the law of dominance is used to explain the expression of only one of the parental characters. Correct? Here only one parental character has been expressed, not the both. But in F2 generation, both were expressed. Okay, so I'll come to that also. And in monohybrid cross in the F1 and the expression in both in F2. So it also explained 3:1 obtained at the F2, and it has also explained the 3:1 ratio in F2 generation. So what is law of dominance? So law of dominance can be defined as in a cross between two pair of contrasting character. Okay, only one of the character is expressed, and that is the dominant character. All right, so this is your law of dominance. Now what is law of segregation? What does segregation mean? Segregation means to separate, right? Okay, now again I'll show you the self-pollination part. See, the self-pollination, what do we got? T, right? Then here during the meiosis, okay, during the formation of gametes, what happened? They got separated. Okay, here what we get to see? We get to see that during the meiosis, the gametes get separated, and in a progeny, both the gametes, okay, from both the parents come, but they do get segregate, okay, but they don't get blend. Okay, if it has got blended, then what would have happened? The result, this would have, the height of the plant should have been what? Average height, right? But as a result, what it is pure, isn't it? Okay, in the first one here you see, if it was blended, then we wouldn't have got this result. Here, though both the gametes are present, but they are not blending. Okay, because of which this gamete has expressed its character in F2 generation. Am I clear? So this T, they are not blending during the segregation. Okay, though they are together, but they are not blended. Okay, so this is called your law of segregation. So dear students, I hope that this is clear to you. What is law of domination and what is law of segregation? So in a law of segregation, if I have to, so here what happened? So two contrasting characters, though they remain together, okay, for a long time, they do not contaminate or they don't blend. Okay, they do not contaminate or they mix or below each other, and they separate or segregate at the time of gametogenesis. It means if a character, if they're remaining together, if they're together for a long time, they don't get blend, they don't get mixed, but at the time of gametogenesis, when see, this is the formation of gamete, right? The gametes are forming, the meiosis is taking place, and they get separated and they express their traits. So this is called your law of segregation. So these are the two laws which was based on monohybrid cross. So in my next class, I shall continue inheritance of two gene, and one more law is there which was proposed by Mendel based on dihybrid cross. So till then, you just go through this video and do write notes on it. Okay, in my next class, I shall continue the next topic. This is all for today. Take care.